Hi everybody, so I'm here on Notability. I have the note sheet that you guys should have downloaded, and what we're going to do is we're going to build a 10 nucleotide version of a DNA strand. Remember that a DNA strand is normally twisted in a double helix, so what we're going to do is we're going to basically untwist it, and we're going to be looking at the ladder structure. Now remember that DNA is made up of nucleotides, and nucleotides have three parts to them. First, they have the sugar, which is deoxyribose. And deoxyribose sugar is where the first part of DNA comes from. I'm going to draw this in blue. I'm going to label at least this first one with the S for sugar. And coming out from that sugar is what I'm going to label as a P. That is my phosphate. And in this case, my phosphate is facing up, almost as if it's raising its hand. So it's going upward at an angle. And to round out my nucleotide, I am going to choose the first of my nitrogen bases, which I am, we're going to use different shapes for this, and I'm going to try and be consistent with the shapes. So I'm going to make this green, and I'm going to say that this guy right here is thymine. So thymine has a particular structure, and the entire thing together, this is all the nucleotide. So this entire piece right here is my nucleotide. Now my nucleotide, remember, is my monomer, and so everything else that I'm going to draw is going to be based on this structure. So I'm going to go back and I'm going to draw my next nucleotide directly underneath my first one. So here is my sugar, and if you remember, my sugar is deoxyribose. It has a phosphate coming out from the deoxyribose going up at an angle, and it is also going to have a nitrogen base, and this nitrogen base is going to be C for cytosine, and hopefully you can tell that that's purple. It's kind of close to its original color. Now these are lined up together, and basically what we're going to have here is a bond between the phosphate and the sugar, and I'm going to make this nice and dark because this bond right here is what's going to hold, basically weld the two nucleotides together. This is a covalent bond. It is the handshake bond that is super strong and for the most part not completely unbreakable, but unlikely to break. So it means that these nucleotides are going to be held together very tightly. So the next thing I'm going to do is I'm going to draw my next nucleotide right underneath. This is, again, my sugar, and I am going to have my phosphate sticking out at the top. And I'm choosing to draw each of my nitrogen bases to begin with here. This is going to be my adenine. However, I could very easily on this have thymine, 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 and that would still be completely legitimate because the order of these nitrogen bases is basically my code. Now I am going to have a very strong covalent bond holding that sugar and that phosphate together, and that, again, I'm continuing my structure here. So I'm going to put another sugar below. So this is my fourth nucleotide. So here's my deoxyribose, and I have my phosphate sticking out at the top, and I am going to have my fourth and final nitrogen base, and my nitrogen base that I'm missing is going to be G, which stands for guanine. My bond between my phosphate and my sugar is a very strong covalent bond holding that together. And I am going to draw one more nucleotide because I said that this was going to be a 10 nucleotide structure. And so here's my phosphate sticking up. And as you guys are doing this, you really do want to make sure that you're thinking about each of the things, structures that you're drawing, because the more that you talk about this out loud, the more likely it is that you're actually going to remember the structure that you're drawing. So here's my covalent bond between my deoxyribose and my phosphate. Now I've drawn one side of the ladder. This one side right here is referred to as a strand. Now, a strand is a strand is basically one side of the molecule. Now, the molecule itself is oriented with a top and a bottom. So the top is right up here. It is I can tell it's the top because the phosphate is sticking up towards the top of the deoxyribose. And this down here at the bottom, at the base of the sugar, is basically what is known as my 5' end. It has to do with the actual carbon that's sticking down here at the bottom. This up here, the top of my molecule, is often referred to as the 3' end. That is referring to the sugar that it's coming off of up here. Now, this is one strand of my molecule, and if you remember, I said that we would be drawing 2 strands, 10 nucleotides, and in order to do this, the easiest way to do it is to basically work from the other side, because I need to make sure that my base right here is attached to the other base on the other side. And to do that, I am going to, again, draw my sugar, but my sugar is going to be facing in this direction, and actually that's not correct. I want to use the blue here to be consistent. So I'm going to use the blue, and here is my sugar. My sugar is attached to the nitrogen base that is complementary to thymine. And complementary means that the shapes are going to have complementary shape and charge. So this is my adenine. Now, in between my thymine and my adenine, we also have bonds, but those bonds are actually weak hydrogen bonds. So I'm going to show those as dotted lines. They are Velcro bonds. Those are hydrogen bonds, the same kinds of bonds that hold water molecules together. So obviously it's not a permanent bond because they can make them and break them. We have, again, our phosphate sticking out towards the top of the deoxyribose. So here I am. I'm going to go with my next nucleotide, and that nucleotide is actually going to be attached to my phosphate. So here's my phosphate here. And again, I'm going to choose the nitrogen base that is complementary to the one that I have in place, which is my G. So my C. So that means guanine is going to go in place. And this guanine is attached to the cytosine using hydrogen bonds. So my next nucleotide, and attached to that, attaching the two of them together between the phosphate and the sugar, is my strong covalent bond. So here is my next sugar. And my sugar has a phosphate sticking out on the upper end. And that is going to now match with, and I have to remember what color, that is going to match with my thymine. So here's my thymine. My thymine is complementary in shape and charge to my adenine, and there are hydrogen bonds holding that together. So here is my next nucleotide. That's going to be a sugar, and it has attached to it a phosphate sticking up top, and it has a nucleotide Let's see, which one would that be? It's going to be our cytosine, which is purple. And so cytosine has complementary shape and charge to the guanine, and it has hydrogen bonding holding it together, and a strong covalent bond, and we missed the strong covalent bond here, so I'm going to add that. So the last nucleotide goes in place. So here is my sugar and my phosphate sticking out on the top. And coming towards the center is my last nitrogen base, which is the complement of adenine, which is thymine. And it is attached using a strong or a weak hydrogen bond. So the weak hydrogen bond, we're just going to label that here. Again, the code is found in the center. That's the order of the nitrogen bases that are in place, and that weak hydrogen bond is going to allow it to zip and unzip when it needs to. And my strong covalent bonds are going to keep my strands together. So you can see that I have my second strand here. This second strand is complementary to the first strand. However, it is running in an opposite direction because the 3' end, or the top of the molecule, is here. I can tell that it's the top because it has this phosphate sticking out, whereas this guy right here, this is the 5' end, this is the bottom, because it is the sugar that is basically sticking out on that end. Now, this idea that one strand runs in one direction and the other strand is going to run in the opposite direction, this is called antiparallel orientation. Oops. And what this basically means is that each strand run in opposite directions. And again, what we're looking at is the open ladder structure, but this would be twisted into a double helix, and that double helix is there to double protect the code. We've got the code tucked on the inside, and then we have it spiraled around, and that creates a double layer of protection. So it's unlikely that something from the outside would be able to damage the code.